That's what I like to hear. You'll be surprised. People do it. They do it. We had a student once who was a good student, took the MCAT, and the very last question of the MCAT, by the way, is the most cruel question you'll ever get. It says, "Do you want to void test? Yes or no?" And this one student almost pressed yes, void the test. But one of the teachers said, "Never ever do that." So I said, "Okay, not going to do that." And then ended up getting, I think it was like a 526, just insane score, and almost voided it. So you really just never know. Cool. All right. I'm assuming there's probably going to be less people today. Some people are in finals week, some people are in the middle of full length tests. So we'll probably just start. Does anybody have any questions or logistical questions? I know this is the final lecture. If you still need to reach out to me, do some office hours, want to meet with me, I'm going to be still available. I know some of you already have. Yeah. You guys are definitely going to have portal access and exam access. If you need that to be extended, I think they're going to expire at the end of January. If you need to have that extended, just email me and it'll get extended for you. And the FLs, no, that's another thing. I think those are going to expire actually at the end of March, the TBR FLs. So if you're taking an MCAT after March for whatever reason, just let me know and I'll try to get your account reactivated. And do office hours end? Yeah, office hours are going to end. Emma might be holding office hours next week. I think she is, but she'll reach out to you guys if she is or not. And then my office hours, if you want to meet with me, I can. And probably Todd's the same way. And yeah. Okay, cool. Let's start talking about biochem lab techniques. Can you guys hear me okay? And can you guys see the screen okay? Yeah, Michaela, I'll meet with you in January, of course. If you need help, especially if you get your test scores are going to hopefully just start going like bouncing back and forth and slowly going up. Oh yeah, and I can send lecture slides. Thanks a lot. Okay, they should be going. Another thing I'm also going to send out to you guys probably right after this lecture is it's like a little study guide. So it's just a list of topics that have seem to be appearing on people's exams in recent times. So it's something that I would recommend going through kind of when you are in between your full length tests, or just kind of twiddling your thumbs, not knowing what to do or what to do that day or that particular moment. It's just like a quick reference sheet just to go through it and do some light brushing up here and there. Obviously the priority for you guys now is full length tests, practice questions as always, and then reviewing everything. And that's what you need to be doing. All right, so for this class, right? It's called biochemistry lab techniques, but the reality is it's biochemistry and molecular cell biology lab techniques, where one begins and one, oh, did that just cut off? Can you guys see my screen? What the heck? Hold on one sec. Is my voice cutting in and out at all? Or is it just the screen share? That is weird. That is weird. All right, hold on. Maybe let's quit out something. Stand by, people. Hmm. Something's letting people. One sec. What the hell? That's not good. Sorry about this, guys. One sec. Return. Yes. Okay, we're back. Nice. So that's a critical piece of advice I have for everybody for the rest of their lives is when something doesn't work, restart it. It just seems to fix everything. Okay, that includes lab equipment, which we're going to go through today. The whenever you're working in lab and something is not working, especially your equipment, turn it off, turn it back on again. It fixes the problem every single time. Okay, so we are doing not only biochemistry, but also molecular cell biology lab techniques, where one of them starts and one of them ends. It's kind of up for debate. And so we're just going to cover both of them. Okay. And so our goals is going to be, we're going to review these very common lab techniques. And then we're also going to look at some very common figures that appear on the MCAT. And then we're going to do some practice questions, of course, related to these various lab techniques. Now, as a recap, kind of, what the this is directly from the AMC, by the way. But just as a reminder, now that we're kind of in the home stretch here, what is the BB section testing at? What is the very general, guidelines that they say they're going to test you? Okay, right? Introductory level biology, some OChem, a little bit of inorganic chemistry, right? You need first semester biochemistry, then cellular and molecular biology topics. So these are all things that we've, gone over now. And so here at the end, it says it's also going to test basic research methods, and statistic concepts. Okay. And so this is kind of what we're going to get at all day today is, the tools that is used in research. And so when you understand them and when they come up in your passages, you'll have an idea of the protocols and what the output is going to be. Okay. So this is what we're going to be looking at a lot today. And again, before we jump into it, it's probably a good idea to just super quickly remind you. I know we've talked about it a little bit before, but how the MCAT is written, okay, specifically the BB section. Okay, we've mentioned that passages, they're very, very commonly adapted from research papers, so published journal articles. They go actually recruit university professors to write the MCAT passages, specifically the BB passages. Okay, and so what you end up getting is MCAT passages that follow more or less this, condensed research paper format. Okay, and within that, for any given passage, you're going to get anywhere from 0 figures, and some of these super passages, which you'll have probably maybe 2 or 3, like I call them double length passages on your MCAT, where you might even get an upwards of 4 figures. Okay, and that's going to include, graphs, tables, charts, diagrams, blah, 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 blah. All right. And so, you can imagine that, a journal article is somewhere like, 7 to 12 pages. Right. And so for the MCAT, when these professors kind of boil it down, it's usually their own research, and they have to boil it down to something like 4 to 7 paragraphs. Okay. And I'm stressing this because in these 4 to 7 paragraphs, that means that they're going to have to fit an insane amount of information, and they're going to use very technical jargon that if you've, taken any full lengths, you're probably familiar with and freaking out about right now. All right. And so if we look at this kind of, condensed research paper format, more or less, it's kind of formulaic. If you do, you kind of see that it's structured in a very similar way for each passage. Okay. And the structure is, follows in some shape or another, right? You're going to have 1 or 3 sentences in the very beginning of your MCAT passage. And that's going to describe, the general topic, right? Maybe the passage is going to be about cancer. It'll say, cancer X is bad, right? And, kind of introduce you into the passage that way. And then that's going to be followed by, 1 to 2 paragraphs describing some sort of, mechanism, some new mechanism that you've probably, maybe are you familiar with, maybe you haven't run into it, but it's going to describe it and it's going to take a few sentences, right? And, back to the cancer example, maybe it's, describing, how a certain cancer is activated. Okay. And then after that, you're going to have something like 1 paragraph or maybe 2 paragraphs that are going to set up an experiment. Okay. It's going to set up an experiment. What are the dependent variables, independent variables? What are we testing? Okay. So like, classic, right? What's the transcription factor? Will overexpression of a certain transcription factor deactivate a cancer? Okay. The paragraph or 2 on that. And then right after that, this is when you're going to get these, figures. Okay. You're going to get, again, 0 to 4 figures. And these are going to, more or less summarize the experimental results. Okay. And so, why is this important? It's because you're going to get tested on the results. Now, if this is the basic layout of each BB MCAT passage, right? What are the very typical types of questions you're going to run into? Okay. You're going to run into one or two questions, typically, generally, right? Ruling of thumb here, that are going to, test you on whether you're able to interpret the results of the experiment set up in the biology passage. Okay. You're also going to get somewhere around, one question that's going to test you on the, specific mechanism and if you can understand that. And then you'll get these, one-off questions. Very common, you'll get tested on something that has to do with the chemical structure, the function of a chemical, right? And then you'll get these, questions that feel like freestanding questions that, don't really have anything to do with the passage. And they just have to do with, whether you know it or not. All right. And so, generally speaking, very generally speaking, what would you say is the most important part of the passage for you to understand? Anybody? Yeah. Well, the results, right? They're not the results. Yes, exactly. The results are going to be given in the figures, right? And you're going to have to kind of interpret the figures to come up with the results, right? Because you can imagine if we go back, right, in a journal article, they're going to have this like discussion conclusion section in a journal article that kind of explain the figures and what they mean. On the MCAT, they don't do that per se, right? They're just going to present the results in the figures, and you're going to have to deduce what the heck went on, and make sense of those results. So I would agree with you, Olap, that the most important, generally speaking, of course, for you to understand is the figures. There's going to be the densest, amount of information in the figures. And so especially if you're, running out of time, the place to look and be able to quickly interpret things is going to be these, the figures and the little notes that they give them. It's also, very important to understand the mechanisms as well, because those are typically the other type of question. But the very hard questions that you're going to get are going to be those 1 to 2, questions that, really test your ability to understand the experiment and the conclusions of it. All right. And so today, that's kind of what I want you to focus on, especially as we're moving into our full length test, moving into, this home stretch. I want you to, focus on, right, what do the figures mean? What do the figures look like? Okay, we're going to rush through a lot of, common lab techniques. And, you need to remember that the lab techniques are, what researchers use to generate, the data. Okay. But, what's presented on the MCAT is going to be the figures. Okay. The figures are going to be, in the background, there's all these lab techniques that were used to generate that data. The figures are what dense information is. Okay. So, it's very critical to understand how each lab technique works, right? But I think it's arguably more critical to understand for any given lab technique, what does that output look like? Okay. What are we measuring? All right. And so let's focus on that today. Okay. Anybody have any questions on that? Cool. All right. And now before we jump in again, just to zoom back out, right. Lab techniques, it's important to review what structures are we even looking at? What are we measuring when we use these lab techniques? Okay. And so, it's probably not a bad idea to review what they are. And this might be an, complete oversimplification, but for the MCAT, we can break the structures that we're looking at into, 4 very general categories, your small molecules, your polynucleotides, your proteins, and your cells. Okay. So, like what is a small molecule, right? That's, like a nucleotide, single, amino acid, single, sugars, alkaloids, glycosides, fatty acids, right? These are examples of small molecules. I think the unofficial cutoff is something that's less than 900 Daltons. Okay. And small molecules are generally, in some way or shape or form, regulate some sort of biological function. Right? What are polynucleotides? That, just genetic information, your DNA and your RNA. There's various lab techniques that look at those. Proteins. Okay. There's proteins are very important. I mean, obviously, it's not a bad idea to kind of go over, right? Here's like examples. There's globular, fibrous, membrane, enzymes, all different types of protein shapes and sizes and functions, right? It might even be worth kind of going through these like globular proteins, right? These are, typically the spherical proteins, hemoglobin. These are often soluble. Something to keep in mind, important when you're doing various techniques. Fibrous is like the opposite, right? These are structural proteins, your keratin, your collagen, these are less soluble. Are globular proteins bigger than usual proteins? I don't know. I'm assuming there's globular proteins of all different sizes, very big ones, probably smaller ones. But you would expect a globular protein to be bigger than say a small peptide. So that would make sense that if I were to take a guess, globular would be in general bigger than things like that. Membrane proteins. This is interesting. This is kind of an interesting fun fact. Membrane proteins make up, I think about a third, if I'm not mistaken, of the genome. The entire genome makes up membrane proteins, which is pretty crazy when you think of it. Okay, these are things that connect cells together. Diffusion, active transport, right? Cellular recognition is for membrane proteins. Hormone receptors, right? Membrane proteins. Okay. Enzymes, obviously, that these are just the catalysts that move reactions forwards. Messenger proteins, right? Proteins involved in like cell signaling, right? Oligomers, right? This just means multiple proteins, multiple subunits, right? So we're going to get into this later, but like quaternary structure. Okay. Okay. What else? Cells, right? What else do lab techniques look at? Lab techniques, different lab protocols, different lab equipment is able to actually visualize and look at cells. So different cell types. Okay. And for your MCAT, when we talk about cell types, there's lots of different cell types, and your MCAT very commonly, something like, I would say, rule of thumb again, maybe 2 to 3 passages will describe the function, or they'll describe rather the dysfunction of any particular cell. Okay, so maybe there's a passage that talks about epidermal cells, right? And maybe a myocyte, which is a muscle cell. Maybe it talks about glial cells. Okay, and so I think it's actually not a bad idea for you guys to familiarize yourself with just because when you run into them on your test, it'll usually describe them and describe their function to you, if a passage is on a particular cell type. But the more knowledge you have going in, the quicker you're going to be able to decipher the passage. If you already have like a background, pre-knowledge, I guess, of what each cell type does. And when I say familiarize yourself, right, don't go overboard. Later, when I send out the this little study guide, there'll be a link on it. And I think that just reading through, there's like a pretty good website I found that has like a pretty nice, brief, basic summary of all the main cell types. And so I think like in between, your tests, maybe just read through that. And you'll get a good idea. Okay. And again, talking about the MCAT, organelles also come up a lot. And so you should, for the MCAT's sake, be able to have a basic understanding of each organelles. So like a typical question would be like, it'll ask for like, where a protein will localize, and literally just choices A, B, C, D will be different organelles. And you'll have to pick, the organelle that makes the most sense for where that particular protein would localize. Okay, so refamiliarize yourself with organelles here going forward. All right. Cool. Okay, so now we know what structures we're looking at, what structures we're measuring when we're using, all these lab techniques. Okay, so now what lab techniques are used for each one? Okay. how do we quantify differences between them? How do we study their function? Okay. If we're looking at small molecules, right, common lab techniques would be, chromatography, column chromatography, ion exchange, size exclusion, affinity, examples. Chemical assays are very common. Okay. Very common lab technique, for measuring the concentration. We'll touch on those in a little bit. Either a colorimetric or a fluorescence chemical assay. Okay. If we're looking at polynucleotides, the big ones, electrophoresis, probably the most common lab technique out there in any lab and on the MCAT. Okay. Especially when you pair it with blotting, which we'll cover. Today, you guys should have already covered cloning and PCR. These also, there's two really good handouts, very detailed handouts for each of these on the class portal. So if you want to rebrush, I would look up those. Okay. But then sequencing, way of measuring this genetic information. Proteins, again, what techniques appear from the MCAT? Electrophoresis, right? But for proteins, very common, the Western blot, which we'll cover. And again, cloning, which would be expression of, an enzymes when I'm referring to cloning for proteins. Okay. And then cells, how do we measure cells? Flow cytometry, okay, is, very, very, it's becoming really powerful. Actually, it's very cool technology, measuring, cells and counting cells. Fluorescent microscope, and then a centrifuge are all techniques around dealing with handling cells, categorizing cells, measuring their function. Okay. Now, I think it's super important to back up one more time. Now we have a bunch of biological structures we're looking at. Now we have a list of the lab techniques that we're, going to study them with. Okay, but the big thing is, what questions are we trying to answer? All right. And so if we wanted to very oversimplify the MCAT and kind of research in general, what are the outputs of whatever lab technique? What are the outputs of those? What are we trying to answer? Okay. So if we're looking at a small molecule, does that particular chemical species, is that going to increase or is it going to decrease in concentration due to whatever experimental change? All right. Similarly, if we're dealing with polynucleotides, if we're dealing with genetic information, right? If we introduce a mutation or maybe some sort of alteration, right? We do some genetic engineering, right? What are we measuring? What do we care about? Does this increase or decrease expression? Okay. These are the questions the very basic questions we're trying to answer. All right. If we're talking about proteins, right? We want to kind of quantify, does protein interact with protein Y? Right? When these two proteins interact, right? Does it increase or decrease the concentration of some other species, whether that's a small molecule or another protein or whatever it might be? All right. Cells, did the physiology of the cell get altered? Okay. And if it did, was it any particular organelle? Did it get disrupted? All right. These are the kind of very basic questions that you should always have in your head when you're going through an MCAT passage. Okay. And so if we were to simplify the very, 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 very high level summary of the BB section. All right. And in some way or another, you're probably going to be asking this question during every passage. It's, kind of boils down to this. Does the increased or decreased concentration of whatever we're talking about, small molecule, polynucleotide, protein, cell, right? Does it cause cellular function to be disrupted or to be resumed? Okay. So this is what you should be looking for in every passage. As you're starting to read through the passage, right? Start to see if you can answer this question. Okay. And that'll help you kind of make sense of everything. Does anybody have questions on that? All right, cool. Yeah, no, let's get into the meat here. Okay. The first one we're going to talk about is chromatography. And you guys might have touched this already in some of your OChem, but it's a lab technique used to separate a mixture. All right. And so these three are just vocabulary terms that you should know, just so when it comes up in a passage, you know what the heck they're talking about. Okay, so you should know what the mobile phase is, right? Which is the mixture that is to be separated. You should know what the stationary phase is, right? So this is going to be the fixed material, which the mobile phase will have different affinities towards, right? So example, the stationary phase would be the resin in a column, which we'll talk about. Okay, and then there's the system, right, where the stationary phase is fixed. So this is going to be like your column, your I forget what it's called, the electrophoresis tool. Gosh, the thing that holds your gel, right? If we're doing electrophoresis, blank in there. Okay. And so for the MCAT sake, when we're talking about column chromatography, there's three that you need to know. You need to know ion exchange chromatography, affinity chromatography, and size exclusion chromatography. I've never heard of these popping up on the MCAT, but the principles are very similar. So if you know these three, you'll probably be able to figure out, hydrophobic interaction or normal phase if it ever were to come up. Okay, so what do we use chromatography for in a lab? What's like the big application if you're working in a biotech lab? Yeah, to separate, in other words, right, to purify. Okay, and so, kind of have in the back of your mind, I've actually worked with lots of columns, and there's columns that are tiny. I've seen columns that, there's like capillary tube columns. I've seen benchtop columns that are, maybe a foot big, and I've seen columns, 3 stories high, just depending on the scale and, where it is, but very common used, in labs and biotech and pharmaceutical companies, right? When they're trying to isolate active ingredients, trying to separate compound mixtures, trying to remove impurities from products, you'll use column chromatography. Okay. And anywhere from an analytical lab to a production facility will have column chromatography. All right. And so the first one, and probably the most common one on the MCAT, would be ion exchange chromatography. Okay, so what is that good for? It's, maybe I'll even write this down. Oh, it's already there. Perfect. Separation of charged or ionizable molecules. Okay, so the most common would be proteins. Okay, which includes peptides, enzymes. You can also do nucleotides, DNA, antibiotics, vitamins, anything that can carry a charge, you can theoretically separate using exchange chromatography. Okay. So how does this work? Okay, the first thing you need to do is you're going to need to pick a molecule. Okay, you're going to pick the molecule that you're trying to isolate. And if it's a positive molecule, so a molecule that carries a positive charge, okay, then you're going to be using a cation exchange, and it's going to have a negative resin. Okay, so this example that you see over here, let's say we were trying to isolate this positively charged protein. Okay, that means we're going to be using a cation exchanger, and that means that our resin, our stationary phase, is going to have to be negatively charged. Okay, so if it was the opposite, if we're trying to look for a negatively charged molecule, then we're going to be using an anion exchange. Okay, then the resin would have to be flipped. It would have to be positively charged. Okay, and so like a very common, just to give you an example of what one of these negatively charged resins would be, in a cation exchange would be like carboxymethylcellulose beads, I think is like the very typical one, right? It's got a pKa of around 1.2. Okay, and so what you're going to do is you're going to put your molecule into a buffer, right? So let's say we have this, you're going to put all of these, your slurry of molecules that you need to separate out, into a buffer. Okay, and remember, the buffer pH should be between the pi of the charged molecule of interest and the pKa of the resin. Okay, So maybe I'll even write that. Of the charged molecule. Okay. And so when you do that, you're going to run your mixture through the column. And in this example here, right, because we're trying to isolate a positively charged, in this case, we'll call it a protein. Okay. You're going to run it through the mixture, and that positively charged protein is going to have an affinity towards those negatively charged beads, these negatively charged resin, and it's going to stick. Okay. And after it sticks, or as it's sticking, everything that's got the opposite charge, right? So everything that's negatively charged here and here are just going to run and they're going to elute out of the column. All right. And so you'll have what's left in the column is just all the positively charged species within the column. Now, what you do next is you're going to apply a salt gradient. And what that means is you're going to start to slowly increase the concentration of salt in a solution that you apply to the top of the column. And as you gradually increase the salt concentration, the positively charged, in this case, because we're talking about a cation exchange, okay, they're going to compete for the positive charges of the protein of interest that you're trying to separate. And eventually, once the salt concentration is high enough, your positively charged proteins are going to start to elute out. Okay. And so the higher the positive charge, the stronger it's going to stick into the column and the longer it'll take to elute out. Okay. And so someone asked a good question. So you want to elute what you don't want, correct? It depends how you design the experiment. But traditionally, when we're talking about columns, right? Let's say I have a protein, let's call it protein X, and it's got a, +5 charge or something like that. Okay. You're going to want this protein that's positively charged to stick to this resin, but you're also going to have proteins that are slightly less charged or slightly more charged, right, within your mixture. Okay. And so as you start to compete, things with different charges are going to elute out at different rates. And you're going to get something called an elution profile, which we'll go over in a sec. But essentially, you'll know where your peak is. And so you'll be able to purify it because they'll separate out at different times. So if you know the time in which your molecule of interest will separate out, then you'll be able to purify it and have it in a different fraction. Okay. So I guess the best way to kind of test this is to do a quick question on it. So maybe take a couple of minutes and try to answer this question here, and then we can go over it, and it should get you to where you need to be. No, and sorry, everybody, someone wants to go back real quick. Just one slide, sorry. Cool. And then I'm going to go forward. Cool. Let's see. Does anybody figure this one out yet? Should be decently straightforward. Got one for A. We got a C. We got a C. Okay, cool. We got a bunch of C's actually. Okay, so what do we do here, right? Actually, first of all, does anybody know what a cell penetrating peptide is? Anybody know what this is? Well, it's exactly what it describes itself as. And it's super cool. Cell penetrating peptide is, if you can imagine the lipid bilayer, right, on a cell. So let's say this is in, this is out of the cell. Okay. Oh, that's a really ugly in. Let me erase that. It's so ugly. Okay. And so you can imagine the surface of the cell has all these phosphate heads, so it's all negative, right? And it's hard for big molecules to get in, right? Because of all the properties of the cell. Now, what a cell penetrating peptide is it's something that has so many positive charges on it. It's a peptide that's got a high concentration of positively charged residues to a point where these negative charges that are on the cell surface will actually attract to these positive charges and kind of fold around them, right? So you can imagine that it creates this kind of micelle to a point where it can actually enter the cell because of the positive charges. So it's one way of getting something to enter the cell, okay, is with these cell penetrating peptides. And so actually a lot of bacteria, they can communicate this way with these cell penetrating peptides, and it'll be a way of communicating between the cells, okay? And so on the MCAT, whenever you see, and these are actually pretty common, whenever you see a bunch of peptides listed out and you see each residue, there's a pretty good chance that you're going to have to count the total charges of each peptide. Okay, so just that would probably be that should be your first instinct. Okay, so if we go down the line and we count these, okay, right, we have one, and I don't think I see any negative charges on any of these. So they only have positively charged residues. So make it a little easier, but 1 plus 1 plus 1 plus 2 plus 3 plus 4 plus 5 plus 6 plus 7. Okay, did I count that right? So this would be plus 7. And then for transportant, we can count it again. Plus 1 plus 2 plus 3 plus 4 plus 5 plus 6 plus 7 plus 8. I think this is right, plus 8. Okay, MGP. Okay, this one has nothing, nothing, nothing. Plus 1, plus 2, plus 3, plus 4, plus 5. This is going to be plus 5. Okay, and then R9 looks like it has 9, so this is plus 9. Okay, so when it says what elution order would you expect? Okay, which one's going to elute out last? Which one of these peptides, right? They all have different charges on them. Which charge is going to elute last? The one with the highest charge. Okay, so R9, because it's so positive, it's got the highest positive charge, it's going to have the strongest affinity for these resins, and so it's going to elute out last. Okay, and so if we're doing our general test taking, right, we just see that R9 is the last one, so we can just immediately cancel out these ones. Okay, we can kind of work backwards here, right? Which one is going to elute out second to last? Transportant, which is this one, and this one isn't. So then the answer is A. Does everyone get that? Okay. Now, another thing Is everyone following this so far? Cool. What if it was an anion exchange? That's a good question. Okay, so if it was an anion exchange, all of these species would be positively charged, so they would all elute out right away. They wouldn't stay with, they wouldn't be in the exchange. And which ones would come first or last? That I wouldn't know. You wouldn't ever design a column to do this, just because it's going to come out in your first wash. And I don't know if it's going to be which one's bigger or smaller, which one is more positive, more negative charge. If in the elution profile, it'll probably be pretty messy. Okay, and you wouldn't ever design your column that way, but that's a good question. Okay, now speaking of this, right? Speaking of, what, because the MCAT might present things in slightly different ways, in unfamiliar ways. What if we were to change this question around, right? And what would the answer choices look like, if we were to graph them on an MCAT, right? So when I talk about an elution profile, right? So the thing that the actual output of a column, okay, you'll have increasing salt concentration, right? And you'll have absorbance, okay? And you'll have these peaks. Okay. And so if I were to ask this question a different way, which one of these peaks corresponds to transportant, which one would you say? Right. And then let's say I made this one A, answer choice A, answer choice B, answer choice C, answer choice D. And just because we already don't want to have to count again, +7, +8, +5, +9. So someone says A. Excuse me. A and C look the same. Do they? Are you sure you know what we're looking at here? Right, I'm asking which peak corresponds to transportin, to this peptide, after running through a cation exchange. Okay, why C? Whoever said C. Second to last peak, exactly. So it's not how big or small the peak is, it's about the elution order. So in this case, the greater the charge, the longer it's going to stick to this resin. So the longer, the more salt concentration, which is another way of viewing it is, you could think of it kind of as time, the longer it'll stay in the column. And so this one will have the highest charge, second highest charge, right? Okay. So this would be the elution order. Does that make sense? Now, for anybody who was tripped up about the absorbance, okay, here's the absorbances. Another question the MCAT could ask is, which one of these molecules did we isolate the most of? Right. So maybe I'm a pharmaceutical company and I had to, pick one of these drugs. Let's pretend they all did the same thing. And I wanted to sell one and I wanted to have the highest yield. Which one of these would have the highest yield? B, exactly. It's the one that has the highest absorbance. This is that biggest signal. The bigger the signal, the higher the concentration. And so this is actually what analytical chemists do in all sorts of bio labs, biotech labs, right? When I used to work in process development and scale up, we would submit our samples over to the analytical chemist and they would run it through, in the analytical chemistry lab through an ion exchanger. And then they would get these peaks and they could, back calculate their peaks and they could tell you how much of each they could give you an estimate, pretty damn close estimate of the concentration of each one within your sample. Okay, and that's how that's how you obtain your KPIs, your key performance indicators, and see if you're doing your job right. Okay. And yes, exactly. So just because it elutes first doesn't mean there's the most of it, right? Your concentration is going to be calculated from your absorbance, right? The bigger the signal, right? And each of these signals corresponds to chemicals with the similar molecules with similar characteristics, aka because we're doing cation exchange, we're looking for charge, right? So everything in here is probably going to have like a +5, +7, right? Okay. Cool. All right. Moving on. Affinity chromatography. Okay. What is affinity chromatography good for? Okay. Affinity chromatography is used to select molecules or enolates based on their biological interactions. So we don't care about charge anymore. It's based on okay, right? How much affinity does have a molecule have for the stationary phase? Okay, right? What's the binding affinity between them? Okay, and so you can imagine when you're designing your column for affinity chromatography, right? That stationary phase, it can be anything, right? It can be some sort of ligand, it can be an ion, it can be a protein, it can be an enzyme, it can be an antibody, right? It can be an antigen, right? You can even have columns where the stationary phase is DNA or RNA, right? Anything that has an affinity towards the molecule of interest that you're trying to purify, all right? And because of this, the big advantage of affinity chromatography is that it's highly, highly, highly selective. If your stationary phase is very specific to the molecule you're trying to purify, then you can have a very, very, very highly selective purification process. The problem when you're actually doing scale up is that it's very expensive. Okay. All right. And so the stationary phase is a ligand that retains molecules based on binding affinity. All right. And so it works very similar to ion exchange. Okay. You have to, pick the molecule that you're trying to isolate. And then based on that, you have to pick the ligand that you want to use as the stationary phase, right? So in this example, let's say we wanted to isolate this famous red triangle protein. I would need to pick a ligand that binds specifically to the red triangle protein. And that's what I'm going to load up my column with as the stationary phase. Okay, and then again, I'm going to put everything into a buffer. Okay, and then I'm going to run it through the column, and then I'm going to wash it with more buffer, and that's going to elute out anything that's non-specific for my stationary phase. Okay, and then once my molecule of interest is bound, if I want to recharge the column, you can add a salt gradient, or also common is you can just add more of the ligand. Okay, and then you can elute out your molecule of interest. And actually, most common in affinity chromatography, it'll still be bound. You don't necessarily need to know this to the ligand, and you'll have to do some further downstream processing to isolate it, to separate them again. Okay. And so very similar process, very highly specific. Now, if you look down here at examples of, the ligand and molecule of interest, like these are very common. I want you to notice his tags, right? And GST tag proteins, all right? Very common on the MCAT, very common on the MCAT. Okay, so does anybody know what a his tag is? Have you guys ran into that yet? Right, his tags are you can actually engineer. You can imagine if you have a peptide, right? N terminus, C terminus. Yeah, histidine tag, exactly. But what does it do? Right. So N terminus. So you can imagine you can actually engineer, you can do some genetic engineering, and you can engineer it so that you have 6 histidine residues on whatever protein of interest that you're expressing. Okay, so you can attach 16 histidine residues. You can even put it right directly into the DNA that gets expressed, makes the mRNA, makes your protein of interest, right? And you can actually get 6 histidine residues. And these histidine residues, right, they have an affinity for nickel. And so if you were to engineer this peptide to have this his tag, you can actually purify that protein of interest because of these histidine residues. And so that's one way when you're running an experiment, right? And you want to be able to purify it to check how much you made, right? To check the presence of it. It's a very common lab technique using affinity chromatography. And same thing with a GST tag protein. You can engineer a glutathione, what's it called? Glutathione S transferase, right? A protein that has this specific region on it that has an affinity for glutathione, which would be the ligand. And so you can purify your protein of interest that way. So yes, so a his tag, right? Let's say I'm running an experiment, right? And let's say I'm interested in some sort of peptide hormone, and I'm interested to see, if my cell can produce it or not, or want to see how much of that protein my cell produces, right? I can do some genetic engineering, and I can attach these his tags to the N terminus of a peptide. And so when I have my cell lysate, so if I purify my sample, right, and I lyse it, and I have all my proteins floating around, and then I put that slurry into an affinity column, right? And I have the affinity column with nickel as my stationary phase, I can purify proteins with his tags, okay? Because those his tags, those histidine residues stick to the nickel, okay? And that way I can, purify that protein of interest. So maybe I'm, in a biotech lab and I'm making insulin, and I want some way to purify insulin from all the mess that's within my sample mixture, right? I can purify it out by engineering the insulin to have this histidine tag in it, okay? And these two are very common. His tags and GST tag proteins, they're just going to be part of the passage, this language in the passage that's describing the experiment. And they're going to say, we used, GST tag proteins, or we used his tags, right? A bell should go off in your head and say, okay, I'm purifying this, right? I'm testing how much I have in my sample. Okay. Cool. So basically, it's like an extra layer of security and knowing. I wouldn't say it's an extra layer of the security. It's just it's a tool. It's a molecular biologist tool. It's a biochemist tool for being able to easily isolate whatever protein that you're interested in studying, right? So if I wanted to study a particular enzyme or something, I can stick a his tag on it, and that way I can purify it later. So now I can, run it through a bunch of different, maybe I'll sequence the protein. Maybe I'll, use it in an assay to test something else, to test some other function. It's a tool for purifying your protein of interest. And you can also get the quantity, right? You can see how much you're making. Okay. Cool. Great questions. Okay, size exclusion. Again, very similar, right? It uses porous beads to separate molecules by their size. Okay. And so the pore size will determine the exclusion limit. And this is particularly used for separating, for purifying molecules of relatively higher molecular weights. Okay. And so, something to keep in mind when you're in a lab, and you don't really need to know this for the MCAT, but you'll have like a series of purification steps. And so like the first step would probably be a size exclusion. So you can narrow, your sample down to something smaller. And then once it's narrowed down, then you would send it through an ion exchange column. And then you might even send it through a affinity column. All right. And so let's just do a quick question on this to see if to kind of hammer it home. Yeah, so maybe take 1 minute, 30 seconds. So again, down here, right, this is our elution profile. Which one of these peaks corresponds to prolactin? I've gotten a couple B's and a couple C's, which is exactly what I wanted to get. And there's a split decision because I didn't tell you which molecules will elute first, right? Do big molecules elute first or small molecules elute first? Yep, exactly. So for size exclusion chromatography, the bigger molecules will elute first. And the idea behind that is, let's say, we have all these molecules of different sizes. If you have a big molecule, it's going to pretty easily pass through. It's not going to get stuck in the beads. Okay. If you have a small molecule like this famous small rectangular protein, right? It's going to be small enough that on its path out, it has the ability to get stuck and actually go inside those pores in each of the beads. And so you can imagine that the path will be much longer. And so the elution time will be much longer. Okay, so the big molecules elute first. Oh yeah, and someone has a cool way of remembering this.
Okay, so my way of remembering this, I did not come up with it. My biochem teacher in college did. He loves McDonald's, so he used this example, but he used the example of going to the McDonald's playhouse with his daughter, who's small. And if he said to his daughter, I'll meet you at the end of the slide, like bottom of the slide, his daughter would want to go through like the whole obstacle course, like the whole jungle gym to get to the bottom of the slide, slide down the slide. Whereas he's a grown man and is too big to do that. So he just walks over to the slide. So he would get there before she does because she has to go through all like the ins and outs of the playhouse. So that's how I've never forgotten it since he told me. So that's how I remember it.
That is funny. You guys got that? So this is the little girl in the playhouse having fun, getting herself in all types of different rooms and the ball machine and all that stuff. And this is your grown ass man trying to get the hell out of there. I like that. Okay, so what's the answer then, right? Right, so this is the biggest. So this will be first, second, third, fourth. Okay, and we're looking at prolactin. So first, second, third, the answer is C. Cool. All right, great. I like that. Okay, chemical assays. You guys, if you've worked in a lab, have probably used so many of these and you're tired of them, but they're just super common in biotech medical labs, right? And there's all different types of assays, but the very, very commonly used to calculate the concentration of any chemical species, aka the analyte. All right. So, for example, you'll use chemical assays to, common, maybe you're calculating the concentration of glucose in the blood. Maybe you're calculating the concentration of ammonia in, a patient's urine, right? You can use chemical assays, these enzymatic kits, to calculate the concentration. And someone wants me to go back real quick to a slide. And, while I went back, just super quickly, I guess, do you guys know what all these hormones do or have any idea? Nice. That's good. The more familiar you are with different hormones, really, the better off you're going to be, because they do pop up. But I will say, things that you need to memorize, I would say, is anything that has to do with the female reproductive system, right? Female reproductive hormones, just pretty much, I feel like they come up on every single MCAT. It's just super reported. Just know your female reproduction hormones. Open the physiology book, go to that section, and just read that chapter. Read those like 2 or 3 pages, and just have a pretty strong grasp on them. Okay. Cool. All right. And so, oftentimes chemical assays, they're performed on, 96 well plates in labs, and you can apply a standard curve to them so you can actually measure the concentration of things. Okay. Whatever chemical you're looking at. And you don't need to know this. This is just kind of to show you how they work. But oftentimes they're paired in a multi-step, two enzymatic reactions, where the product of one reaction will be used in the second reaction. And so for here, if we're looking at this example, we're talking about a glucose enzymatic assay kit. Okay. The idea is that the product of this reaction, which is hydrogen peroxide, is used in the second step of this assay kit, which will turn this chemical species into a different chemical species that just happens to fluoresce. Okay, so you can actually measure the fluorescence of the final product of your chemical assay kit, and using a standard curve, you can actually get, the concentration of, in this case, we're talking about glucose. Okay, so everything will be specific to the thing that you want. You'll have to use a different protocol. On your MCAT, you're not going to have to know any of these protocols. But what you need to know is how this is going to appear on your test, and it's going to appear in an MCAT figure. Okay, so like these are 2 examples of MCAT figures. And glucose concentration, maybe it's insulin concentration. Again, you don't need to know the protocol of a chemical assay, but you should know that it was most likely a chemical assay that, was used to calculate the glucose concentration. Okay, so whenever you're seeing research or a table or a figure, right, and it shows you the concentration of something, just know behind the scenes, probably, if you worked in a lab, you probably understand. If you haven't, just like a chemical assay kit. Okay. So the important part, and, we're going to kind of skip over this, but I want you to be able to, even though there's no passage here, I want you to be able to super quickly be able to look at any figure and have like a pretty good gist of what's going on. Okay. Always immediately you need to just look at the wild type and then see if there's any significant difference between the wild type and whatever you're testing. In this case, this knockout. Okay. And so just practice that, being able to just narrow in on a figure and have like a gist of what's going on. But just to kind of speed things up, let's start talking about electrophoresis. Now, is it safe to say here that everyone has ran gel at some point? Yeah. I hope so. Either, in lab, your internship, high school, maybe your job. This is definitely going to be the most common lab technique on the MCAT, and probably the most common biochemistry tool in any lab. All right. If you were to walk in, you'd probably see different gel baths of all different sizes, with all of these different purposes. And so it's good to know these. And again, you can separate molecules by size. You can separate molecules by shape. You can separate molecules by charge. You can, have different binding affinities. If you use a super specialized gel, it'll actually bind to the gel similar to, your ion exchange column. All right. And so at this point, you probably know this, so we're going to probably just breeze over this. But electrophoresis is used for identifying and isolating, unique proteins, unique DNA, unique RNA sequences, even, and observing changes in the protein's DNA, or RNA, depending on what type of electrophoresis you're running. And the goal is to be able to compare multiple conditions. Okay. So on the MCAT, whenever you see a gel, almost all of the time you might have a ladder if you need to differentiate between the size, but you're going to have different conditions ran in each lane. Okay. And so your goal is to find whichever one is the wild type, or to find whichever one is the control, and then compare it to, all of your changes, right? So delta, whatever. Maybe it's a knockout, maybe it's a knock-in. And observe the differences between the lanes. Okay. And try to think about what the function, what the purpose is of each lane. Okay. And this is just going to take practice and going through practice exams and practice questions to get good at reading gels, and to get good and to understand which conditions are used for each gel, which we're going to talk about in a second. All right. Just to kind of reel it in, there's agarose gels and there's polyacrylamide gels. Which one is used for DNA, an agarose or a polyacrylamide? Most of the time. Yeah, exactly. So it would be an agarose gel. Okay. Oops. Sorry. And I think it's always good to have a general understanding of the cutoffs of certain things. It's just a better way to visualize, your electrophoresis in your gels, right? So agarose gels can resolve anything that's 200 plus kilodaltons, generally speaking, and polyacrylamide, which could also do DNA, but it would have to be very small fragments of DNA, can resolve anything between 5 to 2,000 kilodaltons. Okay, and just for reference, a residue is about 120 daltons versus a base pair, which is 650 daltons. Okay, so that's why you need to use an agarose gel when you're talking about DNA, because you can imagine a DNA running with thousands of base pairs. It's going to get really big really quickly. Okay. All right. Does anybody want to go over how things get separated? I feel like at this point you should know, if it's a bigger molecule, it'll migrate slower. If it's a globular molecule, it'll migrate slower. If it has the higher charge, right? So if it's got a high negative charge, it's going to move quicker to the positive charge. Okay. You guys should probably already know that, so we're just going to move on. What you do need to know is when we're talking about polyacrylamide gel electrophoresis, you need to know 3 main conditions. Oh, someone says, a polar stationary phase will bind a non-polar substance. No, it would be the opposite, right? So if it's polar, it's going to bind to something polar. If something is non-polar, they're going to have less of an affinity towards each other. Yeah, good question. Okay. So for your polyacrylamide gel electrophoresis, or your page, you should be familiar with these 3 main different conditions. You need to be familiar with native page, which is when you run your gel in a native state. Okay, so this will keep the charge of your protein. If we're talking about protein electrophoresis, it'll keep the charge of the protein conserved. So what that means is that you can separate by size, charge, and shape. Okay, and so the subunit interactions within a native cell, those are maintained, okay, which is important because they'll be stuck together and you won't be able to resolve the subunits, but also most of the time with native pages, if you're a good lab person, you can maintain the enzymatic activity by using a native page. So you can actually physically cut out, that piece of the gel, and you can actually purify your proteins and use it, in your further experiments. Okay, there's SDS page, which uses sodium dodecyl sulfate, which is this molecule up here. And what that does is it denatures the protein, which by applying a uniform negative charge. And this allows you to separate by a mass. Okay. So if everything has the same charge, unlike native page, right, which has all three, you can now separate by mass. All right. And so the structures that get disrupted with an SDS page are going to be your secondary, tertiary, quaternary structures. Now, ding, 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 ding. The thing it does not interfere with is it does not reduce disulfide bonds. So it will not denature a disulfide bond. All right. To do that, you need to use a reducing SDS page. All right. So if you run an SDS page gel, and then you also add a reducing agent, the two most common are going to be these two species here, dithioretinol, or even more common, beta-mercaptanethanol, right? That'll not only denature your protein, right, by giving it a uniform negative charge, it'll also reduce the disulfide bonds. Okay. So if you don't already know these, which at this point, I hope you have ran into this before, but maybe take a picture. All right. And we're going to come back to this and have and do a question on this to exemplify it. Okay. Blotting. All right. There's a lot of different blotting techniques. Right. And so why do we blot? Why do we do blotting techniques when we're running a gel? To resolve things. I guess eventually, yeah. So blotting, it's important because let's say I had a sample here, right? I have a sample and it's got, let's say we're talking about proteins. So I pull the sample from my tank or from the body or from whatever it is for my experiment. I've got a bunch of different proteins. So when I run it in my gel, you're going to have different proteins of different masses if we are doing an SDS page, and you're going to get this big streak of different proteins. So how do I find the protein of interest? How do I find what I'm actually looking for? This is when you're going to need to do some sort of blotting. Okay. And if we're looking at proteins, they're going to do something called a Western blot. Okay. And so essentially what blotting does is you can use some sort of radio labeled, antibody or fluorescent thing, or maybe if we're talking about DNA, a hybridization probe specific to our molecule of interest. Okay, so if we're talking about a Western blot, we have an antibody specific to our protein of interest. Okay. And so we can use that to identify a very specific thing. So you can separate it away from this slurry of other proteins that were in your sample, right? So you can prove on your gel, yes, my protein is present or absent, okay? Because I know it's there and I can confirm it, Okay. Very, 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 very common. And so most of the time in your MCAT, you're actually not going to see any gels like this. They're going to have done a Western blot if we're talking about proteins, Southern blot if we're talking about DNA, Northern blot RNA, right? And you'll see these individual bands that have been resolved, proving the presence or the absence of whatever molecule of interest. All right. Uh-oh, did I cut off again? Ah. Did I cut off again? Did my screen disappear? Oh no. Oh no. Shit. Yeah, I don't know what's happening. There's been a lot of rain for anybody not in California. And you know, when rain happens, it just messes everything up. Oh no. Has anybody actually done a Western blot or a Southern blot or a Northern blot? Yes. Nice. Yeah, super, super. They're actually incredibly just frustrating and just kind of hard and finicky. Um, Luckily in the MCAT, it's pretty cut and dry and simple. You know, you'll have each lane and it'll be a different condition and you'll be, you know, used to verify the absence or presence of most of the time a protein or segment of DNA. And you can use that to deduce, you know, what each alteration, whether it worked or not. Okay. We'll go over Southern blotting really quickly. And this process is very similar to Northern blotting and very similar to Western blotting. Okay. But essentially, if we wanted to do a Southern blot, you would take your DNA, you would extract your DNA, and you'd have to do a DNA digestion, right? Which is essentially just cutting the DNA up into little pieces so you can actually run it through your gel. And because we're talking about DNA in this case, you would use an agarose gel. Okay. And so after all your DNA is ran, okay, and you're looking for a specific sequence of DNA, you're going to have to where it is in this gel or whether it's actually present or not. So what you can do is you can use, for Southern blots, you'll have this nitrocellulose membrane. And by capillary action, you'll actually be able to pull up all this genetic information into this nitrocellulose sheet. And then you can incubate it with a DNA hybridization probe, which is essentially a radio-labeled DNA probe that is complementary to your specific DNA sequence. And you'll wait for it to anneal. Okay. And then you can use an X-ray, you know, X-ray autography or some sort of other way of visualization, and you can actually resolve the bands of DNA specific to your hybridization probe. Okay. So this should be reviewed. Does anybody have questions about this though? Okay. Right. And so for the MCAT, what you need to know is you definitely need to know Southern blotting, you need to know Northern blotting, and you need to know Western blotting. Okay. All the other ones haven't popped up, but if you have a good grasp of how each of these work, you'll probably be able to deduce if they try to trick you up with any of these other more technical rare techniques. Okay, great question by Michaela. So you can imagine if here's a piece of DNA, right? Here's DNA, I'm going to do 5' 3' just so you know. You're going to have a radio labeled or fluorescently labeled probe, right? So it'll essentially be the opposite strand. and this will be able to fluorescence if it's a fluorescently labeled probe, or if it's radio labeled, it'll be radio labeled. And so it'll be complementary to your piece of DNA here. And this probe is only going to be 15 to 20 base pairs usually. That's about how long it needs to be able to identify a specific strand of DNA. Just because if you do the math, right, anything that's like 15 to 20 base pairs long, the chances of that sequence being the same somewhere else is like almost impossibly low. So that would be sufficient evidence to say that your piece of DNA is present in the sample. Cool. So this would be your probe, right? And remember, we did a DNA digest, right? So you have lots of DNA floating around in your sample, and then you use restriction enzymes to cut up the DNA so you can actually run it through the gel. So you're actually going to just get, lots of pieces of DNA of different sizes. And you just need to know, find the one that's 15 to 20, that has a 15 to 20 base pair segment of, that'll be sufficient to knowing that your DNA is actually present. Okay. Okay. Here's another kind of like a little cheat sheet, right? So a northern blot, we're looking for RNA and we're using a DNA probe. A southern blot, we're looking for specific DNA and we're using a DNA probe. And then a western blot, we're looking for specific protein and we're using antibodies. Okay. All right, let's kind of do this question. All right, so let's say we take protein X, right, some random protein, and we run it in 3 different conditions. So we do gel electrophoresis under these 3 conditions. Okay, what can we say about each of the structures of protein X? Can we even talk about the primary structure, secondary structure? What can we say about protein X? Can we say anything about the primary structure? Okay, lots of good stuff in here. Why would you say it's big? I wouldn't say, I don't know. There's a ladder here, sure, but we don't even have any numbers here, right? We don't know actually how big it is. So I wouldn't say that we can say the protein is big or not. Right? What is the primary structure, right? That's just the amino acid sequence. Is there anything we can say about that? The only thing that I can think of, we're talking about primary structure, is we know that there's at least one cysteine. Okay, we know that there's at least one cysteine residue. Okay, and that's because under reducing conditions, STS page plus BME, you get these 2 subunits out, which means that, and there was no subunits here, which means that there was at least one disulfide bridge that allows it to resolve these different subunits. Okay. Okay, a ladder is a fixed a ladder is like a control within an experiment when you're running something, whether it's DNA or protein, that you have a known set of different proteins and a known molecular weight, right? So you can actually, the Daltons for each of these. And that allows you to see that if you resolve a protein here, you know how big it is because of the ladder. Right? So let's say this is 100 Daltons. Yeah, this is 100 Daltons because it ran the same as this protein species. Okay. But again, if it's a native page, then you wouldn't be able to tell that because size and shape. Okay. Okay. So someone else said that it's a heterodimer. So how many subunits does protein X have? Are you sure? It might have two, but you could imagine a protein. Let's say a protein has 4 subunits and 2 of them are homodimers, like this, and they're all connected by disulfide bridges. And then you reduce them. These 2 red ones are going to run the same, and those 2 purple ones are going to run the same, right? You're not going to be able to resolve multiple subunits if they're the same size, right? So you got to be very careful. You know that it's a heterodimer at least, and you know that there's at least 2 subunits, but there might be 4, there might be 8, there might be a lot, depending on the subunit composition. Okay. So that's just another sneaky thing that the MCAT might do. All right. Cool. Let's see. We'll do this one. Okay, so let's say this red subunit is 100 Daltons, and let's say this purple subunit is 200 Daltons. Okay. And we do a reducing STS page gel. So all of these are going to linearize. And they're going to separate from each other because we're going to reduce those disulfide bridges. Okay. So now we have 4 different things, 4 different peptides or proteins, and each of these are the same weight. So if these purple ones are 200, right, they'll be stacked right on top of each other. You're not going to be able to tell that there's multiple proteins. You just know that there's 200, right? Even though that there was 4 subunits to this protein. Got it? Cool. Nice. Yeah, we're actually going to skip this. We'll do this one at the end. It's a really good, hard question. But for time's sake, let's move past. Okay. Okay, Sanger sequencing. So there's this legend, this guy called Frederick Sanger, and he was the first guy who really developed the first real DNA sequencing. Okay, and I believe this was in 1977. Okay, and the key to figuring out Sanger sequencing and to kind of understanding how it works, the real key is to actually to understand the differences in nucleotide structure. Okay, and even for the MCAT, I know I've said this before, but they just seem to really like to test on structures of basic biological molecules, including nucleotides and the subtleties between them. So what's the difference between a nucleotide and a nucleoside? You guys remember? Yeah, a phosphate. So a nucleotide, these are all nucleotides. They all have these phosphate functional groups. Now it can be 1, 2, or 3, doesn't matter. If it has a phosphate functional group, it makes it a nucleotide. A nucleoside just has the ribose sugar ring and the nitrogenous base without any phosphates. Okay. And those are, again, free points on the MCAT. Okay. But understanding Sanger sequencing, the crux of it is understanding the differences in different, nucleotide structure. Okay. So quick review, right? A ribonucleoside triphosphate is going to have the hydroxyl on both the 2 and the 3' carbons of that ribose ring. Right. So this will be like something you would find in RNA. In DNA, you find a deoxyribonucleotide triphosphate, where the hydroxyl is removed from the 2' carbon. Okay. And this is going to be abbreviated DNTP. All right. Now, there's this thing called a dideoxynucleotide, and that's when this hydroxyl group is removed from the 2' and the 3' carbons of your ribose ring. And furthermore, when DNA is replicating, it's replicating in the 5' to 3' direction. And in order to attach a new nucleotide, there needs to be a hydroxyl on that 3' carbon. Without it, you won't be able to attach the next nucleotide in the sequence. So if you have a dideoxynucleotide, right, and it gets attached during DNA replication, if another nitrogenous base tries to attach to the next spot, it's not going to happen. And so Frederick Sanger realized this, and he realized that if you perform PCR with dideoxynucleotides, if you mix them in, then the PCR reaction is going to terminate at random points. So whenever DNA polymerase encounters a DDNTP, right, the reaction is going to stop. Okay. And so he super simple, but super, like, just so smart, so cool. He realized that if you have, if you can separate out 4 different reaction tubes, right? So all of them have everything you need for PCR. It has a primer. It has all the DNTPs required for replication. You have the correct DNA polymerase. And then you add one of these DDNTPs, dideoxynucleotides, right? Your A, your T, your C, and your G, and separate those. So each reaction tube only has one of the DDNTPs, that you're going to get termination of PCR at random points in each reaction tube. And so you can imagine, if you run each reaction tube in a gel, you can actually resolve the sequence of DNA based on the size. Okay. So imagine, so what does this mean right here? Something that's resolved this low. This would be the smallest. This would be a DNA that terminated, the quickest. So in this case, immediately on the first nucleotide, right? We had a DDNTP get incorporated. Okay, then it ran the farthest, so it's the smallest. And so based on that, you can actually back calculate what the DNA sequence is based on the size of each DNA fragment that gets ran on the gel. Okay. Does you guys see how this works? Yeah, so from the bottom band is going to refer to the PCR reaction that gets terminated at the very first nucleotide. And then the very next band up will be the next biggest fragment, which is 2 nucleotides, next biggest fragment, 3 nucleotides, next biggest fragment, 4 nucleotides, next 5 nucleotides, right? So you can work backwards and then calculate your actual DNA sequence. So yes. Okay. Super cool. Super, super, super cool. And so this was the first real sequencing. And I believe the Human Genome Project was done this way using Sanger sequencing, and it took forever. I think it cost $1 billion. Yeah, they will. And if they don't say it, this is kind of how you can, they might say they sequence something and you don't even need to know the methodology or the protocol behind Sanger sequencing. You just need to know that it was sequenced. But they have tested and they do want you to know Sanger sequencing specifically, so you can know the nuances of what they really care about is if you understand, how new nucleotides are incorporated. And that's why Sanger sequencing could be tested, is because you need to have a good understanding of the structure and the way DNA replicates. Okay. To answer the questions. All right. And so modern Sanger sequencing is actually still used today. And so first of all, do you guys see all these chemical structures? So these pop up on the MCAT a lot just because they're very big and intimidating looking. If you ever see these chemical structures that are highly conjugated, you should just think fluorescence, right? So these are all fluorophores, right, that you can attach to, in this case, these nucleotides, right, with these prosthetic arms on them that have fluorophores. And so modern Sanger sequencing is really cool because instead of having to do 4 lanes and 4 separate reactions like the original Sanger sequencing, if you can attach a fluorophore to each one of these DDATPs or DDNTPs, sorry, then you can just run them all in the same gel. Okay. And so again, you can see, that the blue, and if you get the blue fluorescence, C, red, G, right? And so again, from smallest all the way up to biggest. All right. And so now the even more modern sequencing, which is used all the time, is instead of having to use a gel, you can actually use a capillary tube that has the same properties of a gel. Okay. And you can actually run super high throughput reads, and generate out each base pair will be read out using a laser, and you get these things called a chromatogram, right? So you can actually sequence, pretty big pieces of DNA really quickly and really cheaply using modern Sanger sequencing. All right. Okay, and we'll do this question at the end as well. This is another really good question. But in the meantime, let's just move on to quantitative sequencing. Okay, so for the MCAT, there's going to be 2 different types of quantitative sequencing methods that you need to be aware of. Okay, there's something called RT-qPCR, which stands for real-time quantitative PCR. And then there's going to be RNA-seq. All right. Real-time qPCR quantifies the transcription of known genes. Does anybody know why they have to be known? To do any PCR, you need primers. Okay. So to do any PCR, you need primers. And the only way you can use primers are if you know what you're looking for. Yeah. I got you, Sophie. I knew that was you meant. Okay. So for real-time qPCR, you need to know what you're looking for, but it'll allow you to quantify, how much of that gene you're expressing. Okay. Versus RNA-seq, which is a relatively modern and incredibly sophisticated and honestly cutting edge technique used to look at whole profiles of genes within a sample and determine which ones are active, which ones are being expressed more, and how much they're transcribed. Okay, so RNA-seq is very useful for searching the entire transcriptome. Okay, both are very useful to compare 2 populations of cells. Okay, and then between those 2 populations, measure the actual differences in gene expression. All right. So, you can imagine on your MCAT, you're going to get your control or your normal set of cells, And then you're going to have a set of cells that are, they'll describe it, but they'll either be mutated. Maybe they were grown under different physiological environments. Maybe this cell line was genetically engineered. And you're going to want to compare between those two cell populations. Okay, you want to know, what did this mutation do? Are we making more or less of this gene of interest? All right. And so you can use real-time PCR to be able to do that and to compare them. Okay. So when you do PCR, you can imagine PCR, you're amplifying the amount of DNA present, right? So you can imagine that you have a really small starting material, then all of a sudden you hit this exponential phase, and then it kind of levels off. This is your PCR reaction. Okay. Number of cycles. So what real-time qPCR does is you'll have your two samples. So let's say this is, wild type, and let's say this is overexpression or something. Okay. So you want to be able to measure how much your sample, your mutated cell line is overexpressing this gene of interest, YFG, versus the wild type. Okay, let's make it this gene right here. So real-time qPCR, right? First, you let your cells express the mRNA. And then in the protocol, you turn that mRNA into cDNA, copy DNA. And then the copy DNA, you do qPCR. All right. And if you were to do this for both your wild type and your cell line of interest, okay. So again, mRNA, cDNA, and then your PCR, right? If you have more starting material, AKA if you have more starting mRNA, and you're comparing these two, which one is going to hit exponential phase in your PCR quicker? The one with more starting RNA or the one with less starting RNA, mRNA? Exactly, right? So if you start with more, you're going to hit this real time, you're going to hit exponential phase sooner than if you have less starting material, even though the end product is the same. Okay. And so what real-time PCR takes advantage of is it uses PCR with this specific with these probes, right? So you can imagine on this copy DNA, you're going to incorporate this probe, this DNA probe that has a reporter, so something that fluoresces, and a quencher. Now, all a quencher is that when it's near a reporter, it quenches the fluorescence, okay? And so it takes advantage of DNA polymerase, which as it's building new DNA, you have to remember that DNA polymerase also acts as an exonuclease. So if there's any DNA that's been annealed, AKA this probe that I'm talking about, it'll chop it up. And when it chops it up, it separates the reporter from the quencher, and you get this fluorescent signal. So as you're doing your PCR reaction, okay, all of a sudden you're going to start to detect a signal the second you get into your exponential phase. And you're going to hit your exponential phase quicker if you have more starting material. Okay. And if you use a standard, right? So it's like a control, like a standard, something that you have a known amount of starting material. you can actually back calculate how much starting material you had between your samples. So you can get an actual real number, and it'll come off as like nanograms per liter mRNA. And so the higher your nanograms per liter, the higher your expression levels. Okay. And that way you can actually see what each cell line is doing. I know this is pretty complicated stuff. Does anybody have any questions on it?
I have a question, Josh.
Yes.
Like say if they're using this for a COVID-19 test, do they take like the average of the two sides?
Absolutely. So this is great. So when you're talking about a COVID-19 test, right, you are declared positive for COVID if you have a high enough starting material. So they'll actually do real-time qPCR. Okay. So for instance, there's, let's say they've actually, the CDC, or I don't know, whatever organization who put the guidelines out has actually changed them. In the beginning of the pandemic, right, if you had a certain amount after, I think it was like 40 cycles, then you would be considered positive for COVID. And so they actually over-reported the amount of people positive for COVID. They've actually lowered the threshold of starting material, right? Because with qPCR, if you have a little bit of, you know, the COVID sequence, right, the DNA sequence, you know, in the COVID virus, you're considered not positive if you have so low of it that it can't be detected in, you know, whatever, I think it's now 20 cycles of PCR or something like that. Does that answer your question, or were you asking something else?
I think just like, do they take the average of the two, or like if it's exponential, like you for sure have it, or like, I guess like how do they differentiate between, I don't know
Between
how to ask it.
How they would differentiate between I think they're just saying, okay, so right, when you amplify DNA via PCR, you can see if it's present or not. Okay. Yeah, exactly. So what Sophia said, right? So PCR will just allow you to say if it's present or not. Now, if you do quantitative PCR, it'll tell you how much you started with. So if you start off with a very small amount of COVID, then you're considered not positive. Yeah, exactly. It doesn't detect if the DNA is alive or not. It just detects the presence. So you can imagine if, even if you have the tiniest amount of DNA and you amplify it like crazy, right? 40 cycles, you're going to get a signal from that. If you don't amplify it, if you amplify it less, then you're not going to get a signal. Yeah, super cool stuff with COVID. And honestly, I don't know if it's probably not going to come up on your MCAT. COVID is probably going to be another year or two before it actually reaches the MCAT. But don't be surprised, right? But real-time quantitative PCR allows you to back calculate from your standard your actual starting material. So you can actually quantify the amount of mRNA expression, and you'll get an output like nanograms per liter. Okay, RNA-seq, you definitely don't need to know the protocol on the MCAT, but you should know the output. Again, it's measuring expression levels. So it's measuring the amount of transcript pulled out of a sample. Yeah, this thing is called a heat map, right? So with RNA-seq, it's super cool because you can search the entire transcriptome, right? So you can look at all these different genes at the exact same time in all these different cell lines. And then you can see which ones are more or less expressed compared to each other. Super, super powerful. This is how you find, this is like, there's lots, this is in, you know, biolabs all over the place, you know, looking for targets for genetic engineering to like improve a strain. For example, if you have like a cell line that's making like a drug or something, look for bottlenecks in the metabolism, but we won't go there. For RNA-seq, what you should know is what the output would look like. And again, it's just going to be for comparing to expression levels. So you might say, okay, you might have a table that says expression of this genetic line has higher expression than this genetic line. Okay. And so if you see RNA-seq, that's what it's comparing. Okay. And, while it's good to, again, to know how these lab techniques work, the most important thing is to be able to understand and read an MCAT figure. All right. So here's, just examples of how this might be presented to you. Okay. If you're talking about mRNA expression, chances are they used RT-qPCR or even RNA-seq to generate these numbers. And so most of the time, you don't even need to know the protocol or the specifics of it. You just need to, if they use real-time qPCR, that's what's generating this data, right? Relative expression of this gene, right? Real-time qPCR was using that. Okay. Okay, figure 3 here. If you look at the y-axis, relative number of red blood cells, how would we do that? What technique is used to probably use to count the number of red blood cells? Flow cytometry. All right. So flow cytometers, it's a relatively high throughput method for measuring single cells. The cool part here, one cell at a time. High throughput. So you can imagine what a flow cytometer is you'll take your sample and it'll have all sorts of different types of cells, maybe, or maybe it's the same type of cell, whatever it is, and it'll funnel it down. Yes, this is how they do exactly, Sophie. This is how they do a blood count. When you go get your blood drawn at the doctor's office, they'll use a flow cytometer. And there's different ones. There's some that are super sophisticated, some that are, pretty crude. But the idea is that you funnel the sample of cells through a tube that's small enough to go single file. So it's one cell at a time. And as the cells are passing, you have a laser that hits the cell and it'll create this diffraction pattern and it'll hit a detector on the backside. Okay. And so you can imagine if a cell is different from another cell, then you're going to get these different light scattering patterns that are going to be detected differently by the detector. Okay. So this is why if you have two different populations of cells, maybe red blood cells, white blood cells, maybe white blood cells and a cancerous white blood cell, okay, you can actually use the different deflection patterns to quantify those differences. Okay, so you can actually get a specific cell count and you can look for specific types of cells. Okay, the super fancy ones can pick up, well, obviously imagine if you had like an antibody, a fluorescent antibody, right? And you wanted to look for specific structure within a cell. You can have that antibody bind to the specific structures in a cell and then send that cell through flow cytometry. And so you can get the actual count of cells that have that particular structure present using a flow cytometer. So like the super common application is you can see difference between leukocytes in your blood, right? So this is people with leukemia, they'll go in and get their blood drawn all the time, and you can use a stain on the DNA and pass it through a flow cytometer that can identify cells with more total DNA, which means they're cancerous, and so you can actually track the progression or regression of the disease. Okay, so super cool stuff, flow cytometers. Okay, I think this will be a cool one to do a quick question on. Yeah, maybe take a few minutes, 3 or 4 minutes to do this question. Yeah, exactly. Marissa. No, I mean, if we're talking about like leukemia specifically, yes, that's probably what they're talking about is, the total, like the concentration of cancerous cells within the sample, right? To see if the chemotherapy is working or not. Of course. Oh, anybody have an answer for question 2? Anybody else? Yeah, these ones are hard. This is I really like this question for a lot of reasons. At least question 2, specifically because as you recall, most of the graphs you run into, in a lot of practice examples, practice questions, have the fluorescence intensity on the y-axis. This graph is a total head spin because it has the fluorescence intensity on the x-axis. Okay. And so to understand this question is we have to understand what does the fluorescent intensity mean? When you look at this graph, what does this mean? As we go this way on the graph, what does that mean? Not more numbers of cells, right? This means higher concentration of DNA. Okay. So like what this flow cytometer is doing is it's using a dye to bind to the DNA, and then it's measuring the fluorescence intensity. The laser is measuring the intensity of that DNA. Okay. So the higher the fluorescence signal corresponds to the more amount of DNA. And so the higher if there's more DNA, right, that is an indicator of what stage of the cell cycle you're in. Okay. So if we do a quick review of the cell cycle, okay, if you're in the G1 phase, the growth one phase, right, you've got one copy of DNA. If you're in the S phase, that means that your DNA is actively replicating. So you're actively increasing the amount of DNA in your cell. And then once you're in the G2 phase, you've got double the DNA. Okay. And so if you recall this, when you go back to your graph here, you can use that to differentiate which stage of the cell cycle each one is in. Okay. So the answer to this is C. Do you see that? So from this region to this region. And that's because you're getting cells. Oh, shit. Thing cut off. I don't know what the heck. Yeah, I say that. Let's see if I can get this quickly. Sorry. I don't know what the hell's going on. Nice. Oh, good. Okay. So you guys see how the S phase is region C? Yeah, of course. So the fluorescent signal, you get a higher signal the more DNA you have. Okay. So the higher the signal, it's picking up more DNA. And remember, we're using a flow cytometer, so we're measuring each cell one cell at a time. Okay. So each time you get a signal, right, it's measuring a cell, and the higher the intensity means that cell has more DNA if the signal's higher. So now if we review the cell cycle, right, if you have to remember that G1 has only one copy of DNA. The S phase is when DNA is replicating. So it's going to go from having one copy to two copies. So it's not going to have a peak. It's going to be somewhere in between one copy and two copies as it increases. Okay. And then once you're in the G2 phase, you're going to have two copies. So if you're looking at the flow cytometer, the output, right, you can imagine, and I guess we're going to get into question 3 now, right, that D would correspond to the G2 phase, right? C would correspond to the S phase, right? B would be, G1. Okay. Does that make sense? Yep. Exactly, Sophie. It is because it has more DNA than G1. And yes, exactly. So you can imagine, right? All the cells in here that get recorded right, you're going to have varying levels of intensity because all the population of cells in here are all replicating, right? They're going from having one copy to having two copies. And so depending on what's, some will be just starting replication, some will be just finishing replication. But you'll see this, that's why there's, such a wide range of fluorescence intensity there. Because it's not picking up any DNA. It's not picking, I guess it is, yeah. Why is G1 not A and G2 not B? And S is C. Well, I'm not understanding. Why is G1 not A? I guess just because most of your cells are going to be in G1 most of the time, right? In a given sample. If S has more DNA than G1 and G2, S does not have more DNA than G2, right? Yeah. Okay. This is just saying that these cells are replicating because it has more DNA than G1, right? But not as much DNA as G2. Yeah. Nice, Marissa. Yeah. Does that make sense? And so what's the answer for number 3? Yeah, exactly, G1. And that's because here we're looking at cell counts, right? So if you take the area under this, that'll give you the total number of cells in G1, right? Yeah. And so this is a very good question because it's really making you not, every time on your MCAT, look at your figures, right? Look for your wild type or your control. And if that's not there, or if it is there, you always got to look at your X and your Y axis. Oh man, is this so slow? There we go. Okay. And because the MCAT is the king at presenting things in a slightly different way than you're used to. You know the answer, but they present it in a way that feels unfamiliar. Yeah. Cool. All right, let's just quickly touch on differential centrifugation. Super simple. Right, differential centrifugation it's a technique used to separate different cellular components. Okay, so the idea behind it is different cellular components have different densities. So you could imagine that a nucleus is going to be more dense, than like a microsome or something, or fragments of an ER. And so based on each part of a cell's density, and depending on how fast you spin it in a centrifuge, you can pellet them out at different gravities, right? Different RPMs, if you will. Okay. So you don't have to memorize this, and you don't have to memorize the actual these would be like protocols, like very general guidelines. You should have a very rough understanding of what will pellet before something else. Okay. So you should know that tissue homogenate will pellet out at very low speeds, right? And same, with like nuclei, whole cells, okay, those will pellet out much quicker than, say, a microsome will pellet out. And at the very end, your supernatant, after everything has been spun down at incredibly high speeds, the last thing is going to be those soluble proteins I was talking about. Okay. So pretty basic, pretty simple, but just I would be familiar with what would pellet out first, the general order of things pelleting out, because it does pop up. Okay. The similar thing would be a sucrose density centrifugation, which instead of spinning at different speeds, you can apply your sample into a tube that has a sucrose gradient. Okay. And then when you spin it, depending on the density, your different parts of your cell sample will be able to move further down or remain up high in the gradient. Okay. So the denser, the lower it'll be in a sucrose density centrifugation. Yeah, but based on density. So kind of like size, but right, mass over volume. Yeah, and so this, you can imagine here, like this would be your microsome and this would be your nuclei or something like that, depending on when it will centrifuge out. Okay, does that make sense? Cool. Okay, lastly, we'll talk about, or second to lastly, we'll talk about immunoprecipitation. Okay, and that's pretty much exactly what it says. It's precipitating something out of protein using an antibody. Okay, so you can imagine if you have something that's soluble, right? So it's an aqueous, and let's say it's a globular protein that's soluble, and you want to study it, okay, and you want to pellet it out. You need to make it insoluble, so it's not the supernatant. So what you can actually do is you can attach antibodies, or sometimes it's a secondary antibody, and these can be attached to insoluble little microbeads. Okay, and once it attaches to this bead, the protein will stick to it via the antibodies, and then if you spin it down in a centrifuge, it'll actually pellet out. Okay. And so two very common methods, techniques that kind of use this basis would be your chromatin immunoprecipitation, your CHIP. And there's also something called a protein complex immunoprecipitation. Okay. Chromatin immunoprecipitation is precipitating protein and DNA complexes. Okay. So a very common application of this would be if you wanted to, identify the location on your DNA. So the sequence of DNA that corresponds to a particular protein binding site. All right. And this down here shows that. So if you have your DNA and you want to know where a particular protein will bind, you can incubate your proteins with the DNA, right? You can chop them up. You'll have these little fragments of DNA, and some of them will have that protein bound to the DNA of interest. Okay, then you can use your antibodies or your secondary antibodies, depending on what it is, to bind to that protein that's bound to the DNA. And then you can do your immunoprecipitation, and then you can wash it, and you'll have your protein bound to your DNA, and then you can reverse cross-link it. And then you'll have these DNA fragments, these short DNA fragments that correspond to the sequence of DNA. Oh, my thing went out again. Of your DNA, of your protein binding site. Okay, so this is like a super common thing to look at, like a transcription factor. if you knew a DNA sequence, or I mean, if you wanted to find the DNA sequence that corresponds to where, like, say, a transcription factor binds, then you can use CHIP. All right, let me try to turn this on. I'm sorry about that. Okay, cool. That was quick. Right. And so, like, protein complex immunoprecipitation, right? Let's say you had a known protein that forms a complex to be active or inactive, whatever it is, and you want to find out the other proteins associated with it. You can bind your protein, mix it with the proteins that you're trying to find, and then immunoprecipitate them out, and then actually study them. Okay. Any questions on immunoprecipitation or applications? Yeah, absolutely. So in a lab, here's your sample tube. And let's say you pull a sample. Let's say you grow a cell culture. So you grow a cell culture in a flask, and it's a bunch of cells that you're interested or you're studying. In your sample, right, it's just going to be this thick black sludge of everything. Okay, so this is going to be all these cells, nutrients that are in the media, whatever it is. It's all going to be kind of mixed and brown and sludgy. And when you do a centrifuge, right, it spins it around really fast, which pushes everything that's got density down into the bottom of the tube. So if we look over here now, after centrifugation, you're going to have a pellet at the bottom of the tube. And everything above it, if you spin it fast enough, is going to be what's called supernatant. Okay. And so everything that's left in the supernatant is going to be things that are insoluble or completely soluble. Like proteins and things left over that are not in the pellet, which is going to be, cells, things that are heavy.
Wait, sorry, can I just ask?
Yeah.
So if you were centrifuging like a cell and it was broken apart, so like a full cell would be in the pellet, but like the organelles and stuff would be in the supernatant. Is that idea?
Exactly. So like imagine, so like I worked in a fermentation lab. I'm a fermentation scientist. So I'll go to one of my tanks and I'll pull a sample, and that'll be a bunch of whole cells mixed in broth, right? So in everything that in the broth, if it's certain cells will excrete things, so that'll be in the broth, but then you'll also have whole cells. And so when you spin it down, the whole cells will fall to the bottom of the tube, right? So this would be, in this case, whole cells. And then everything in the supernatant would be what my cells have excreted during my fermentation, right? So what is left the cell. Okay. You can also lyse your cells, which is in a lot of protocols. So you have whole cells and you're interested in something inside of the cell. You can lyse it using a chemical, and now all the cells bursts. And now everything that's in the cells will be out in the broth. And then when you spin it down, right, depending on how fast you spin it, if we go back to differential centrifugation, you can keep things in the aqueous solution depending on how fast you spin it. Okay? Does that make sense? So maybe let's say you wanted to isolate the mitochondria, you would lyse the cells and then only spin it at 80,000 Gs. And that way all the other cell debris will pellet, but it'll leave behind the mitochondria and everything else below it. So it'll leave up behind all this stuff. And this is the first thing you do in every lab technique ever in biology, is you take a sample and then you spin it, and then you try to isolate the thing that you're actually interested in studying, right? Because you know where it's located, right? And so this is like the very first thing you do everywhere, every lab technique. If you spin it at 80,000 Gs, yes. You spin it at 80,000 Gs, anything below will be in it. If you spin it at 20,000 Gs, I'll put it in red, it'll be everything here below. Okay, if you spin it at 150,000 Gs, and you don't need to memorize these numbers, but you should just kind of memorize, not memorize, but just have a general idea of which ones are more dense or less dense. Right?
So if we're to take your example, so like if you wanted just the mitochondria, and then you spun it at 80,000, then the pellet would contain the mitochondria and like what's below it, and then everything else would be in the supernatant.
Oh yeah, sorry, maybe I had that. I was
Or is
thinking
it
this.
separate?
So if you spin it at 80,000 Gs, right?
Mhm.
Pellet contains mitochondria, so it'll contain everything up here.
Okay,
Right.
so the pellet would contain what it is that you're looking for.
Yes and no. Depends on the protocol you're using. Sometimes you're interested in what's in the pellet. Sometimes you're interested in what's in the supernatant. But
Okay,
this will be
got
the
it.
cutoff, right? So everything in the pellet will be this way. Everything in the supernatant will be this way. So if you wanted to study the small vesicles, then you would run your gel based on things in the supernatant. If you wanted to study the mitochondria, right, then you might take the pellet and then downstream process it and separate things out, yada yada yada. Okay, but that's just it's the cutoff line between what's in the pellet and what's in the supernatant.
Okay, got it. Thank you.
Yeah. So yeah, exactly. Cool. So you can get rid of each half, whether it's upstream or downstream, and then use that to help purify what you're actually looking for, actually trying to test in another, biochem, biotech, whatever it is, lab technique. Okay, I hope that answers that. Immunoprecipitation, any questions on that? Cool. You can always ask him a little bit. Lastly, fluorescent microscope, which has popped up on the MCAT. You don't need to know the mechanism, but it's kind of good to get an idea of what it is. All right. So fluorescent microscopes, by the way, it's a super cool way to get a live look at cells. Okay, it's very useful because you can see, what's going on to a specific structure within the cell in real time. The cell can be alive and functioning, and you can actually visualize it using a fluorescent microscope. Okay, so like maybe you want to, look at to see if a particular membrane protein is getting redirected to some other part in the cell, right? Which would be a dysfunction, right? You can actually use the fluorescent microscope to visualize that. Okay. And the way it works is you have light, right? So your light source gets passed through this thing called an excitation filter, and then it bounces off this thing called a dichroic mirror. Okay. Oh, sorry. The excitation filter filters out all light except for light of a certain wavelength. So in this case, it'll let this green light in, which will then bounce off a dichroic mirror, and a dichroic mirror reflects light of a high frequency, but lets light of a low frequency through. Okay, so green, which has a higher frequency, in this case of red, will hit the dichroic mirror. It'll bounce off. It'll hit your sample. When you hit a sample, it'll change the frequency of the light that reflects off, which will be lower. So it'll allow it to pass through the dichroic mirror and then up into an emission filter, which just cuts out noise, and then into the camera so you can take a picture. And so whenever you see a fluorescent microscope, one of those beautiful pictures here, just note that these are 3 pictures overlaid on top of each other because you can only do one light at a time, but it's probably like light, and then, snaps this image. Okay, so depending on the structures that you stain, right, you can apply different stains that will fluoresce different colors. And so you can actually visualize them in real time in your cells. Okay. Right. And so for the MCAT, what's literally been a question is I've seen a picture of different stages of the cell cycle that underwent fluorescent microscope, and you just had to identify, it'll say, what stage of the cell cycle is that one based on the fluorescent microscope image. And so just, again, I can't stress enough, you should have a pretty decent understanding of the cell cycle just because it's so common. Okay. And that's it for those. I will do questions. I think we should go over those questions if you guys want to stick around. But before we do that, does anybody have any questions about any of the material or any questions about, yes. So host DNA, let's say you have DNA, right? Host dye is a blue dye or reflect blue, right? That will bind to, I think it's the major or minor groove of DNA. So it's a chemical that essentially binds to DNA. And so it'll stain it blue. So you can visualize it on a fluorescent microscope, right? And you'll get these blue structures. So it allows you to visualize DNA. Does that explain your, right? So it'll be actually this blue here, right? This is looking at DNA. Cool. Yeah, awesome. Any more questions about any of the material in this? I know we had to move pretty fast. So I can go over anything again, and then if there's no big questions, we can do those other that other practice question. I think there's two actually. Would a silica gel be a polar stationary phase? I would say so, yeah. But what's the context of that question, Marissa? Was this like a homework question?
Oh no, I think I've just seen like questions where they just give you like, oh, like a silica gel is used to separate like these two things based on the structures, like these two different structures, what would have like a higher RF and what would have a lower RF, that type of thing. So then you would just assume that like based on what type of gel it is, you would have to figure out what is the polar stationary phase or,
Yeah,
or what's the
exactly.
stationary phase. So I just wanted to make
Yep.
sure if silica is polar or considered to be polar.
I would have to look. I would assume so, just
Okay.
based on the molecule. But yeah, I've never seen a question like that, but I can see that coming up, right? And it's, what are you trying to measure? What are you trying to, just think about it as like in the column chromatography, right? That kind of concept kind of applies to anything like that. So yes, I would say yes.
Okay, thank you.
Yeah. Cool. And then I'll leave this up and then we can go over it in like 3 or 4 minutes. It's a little tricky question, but it's good. Cool, we got one. I know this is a hard one. Very dense. We got another one. The answer. Hold on. Nice. Okay, we've got one of each answer besides D. We got one of this one, one of this one, one of this one. Okay, so with a question like this, it gets really just, you can get really quickly disoriented just reading through all the dense lingo and technical jargon. And so when I, approach things like this, I try to simplify the question as much as I can. Okay. So you read the question one, based on the results in figure one. Okay, here's figure one. Would the use of a calcium channel blocker be a potential pharmaceutical target for treating patients with Parkinson's disease? Okay, so I want to simplify that. Okay. I know we're looking at, calcium concentrations. And so I'm going to rewrite this to say, calcium channel blocker, what does that even do? That lowers the calcium. Does lowering the calcium concentration, lower Parkinson's disease, right? Parkinson's. Okay. So now I'm looking for, what does calcium concentration do? All right, in the passage. Okay. And so you'll see the gel here. First of all, this is also very classic. Like, what the heck is this? Again, you need to get into the habit of simplifying things. Okay. So you're like, what is this? It's increasing in concentration. Okay. Right. So you'll read to test the effect of intracellular calcium, blah, 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 blah. For 8 hours in media containing a 0 to 10 micromolar calcium ionophore, and then they give you the name. So essentially, this is just saying calcium concentration. Do you guys see that? How I'm just crossing things out and simplifying them down to what I know they are. So, right, exactly. Okay. And so, okay, phosphorylated synuclein, right, at serine residue 129. Okay, what does it mean when the bands are getting darker? More concentration? Yep, exactly. So if we're increasing the calcium concentration, AKA more calcium ionophore, we notice that the phosphorylated alpha-synuclein concentration goes up. So you increase calcium equals increase in phosphorylated alpha-synuclein. Okay. And so what does, right, every time you have a different species, it's good to, like, you always are looking for the function, right? What does this do? And that's when we're going to go into our passage, and you guys have probably already read it, but I need to find where to highlight. These diseases are manifested by internal containing high abundance of phosphorylated alpha-synuclein. Okay, so higher phosphorylated alpha-synuclein is bad. So our goal is to lower the amount of alpha-synuclein. And so if we work backwards, we can see that lowering the calcium concentration, right, is going to help us lower Parkinson's disease. All right, so which one of these answers satisfies all of those parameters? Right, let's see it. One, yes, elevated intracellular calcium showed an increase in phosphorylated alpha-synuclein, which leads to increased oligomerization and neurotoxicity. That actually sounds pretty good, doesn't it? It also happens to be the answer. Okay. Elevated intracellular calcium showed an increase in phosphorylated alpha-synuclein, which leads to a decreased oligomerization and neurotoxicity. No, it leads to an increased, right? No, yes, elevated. There's showed a decrease in phosphorylated alpha-synuclein. No, that's the opposite of what the gel is telling us. And no, elevated intracellular calcium showed a decrease in phosphorylated alpha-synuclein. No, and that's the same thing. We can cancel that out. So Monica says, so I thought phosphorylation was good. Phosphorylation of alpha-synuclein has been reported to occur at several residues, including serines 87, 127. Oligomerization and neurotoxicity of alpha-synuclein can be regulated by phosphorylation. Right? So it's regulated by phosphorylation, but that doesn't tell us if phosphorylation is good or bad. And this is going to pop up actually a lot on your MCAT and your MCAT practice exams. Proteins and enzymes get phosphorylated, and I want this to stick, right? It turns proteins on or off, right? It makes something functional or dysfunctional, depending, and sometimes phosphorylation will turn it on, sometimes phosphorylation will turn it off, okay? And so every time you're dealing with, you know, a passage that talks about phosphorylation, yeah, exactly. So how do you know A versus, because of the sentence I highlighted here, these diseases are manifested by the intraneural Lewy bodies containing high abundance of phosphorylated alpha-synuclein. So these Lewy body samples show more phosphorylated alpha-synuclein. Lewy bodies are bad, right? Parkinson's disease and several other neuronucleopaths. These diseases are manifested by these Lewy bodies containing high abundance of phosphorylated alpha-synuclein. Does that make sense? Does that answer? Does that clear that up? Okay. So yeah, tricky, jargony, right? You know, what is this lane right here? What does this serve? Control, exactly, right? So if these were getting lighter or darker, then you would know that the experiment is a bust, because then these numbers wouldn't mean anything, right? But the fact that the phosphorylated alpha-synuclein is getting darker and the total alpha-synuclein is the same, it's a control for this one. And that's the same thing as beta-actin. So beta-actin, and again, MCAT's going to throw things that you just have no idea what they are, right? But you just need to get good at being like, I don't need this information. This information might be just a control. But if you knew what beta-actin is, it's something you load into the wells, so you know you've loaded the same amount into each well. So it's another control. Yeah, exactly. Monica, thank you. Yeah. So negative control is if nothing were to happen. Yeah. So if you have a negative control and something pops up, then you know your experiment's bust. Okay, so takeaways here: simplify the question, right? Simplify your figures, and always make a summary of like what's going on, right? You know, like a one sentence, oh yeah, I was right here, right? This thing just equals this. And then once you know what's happening in the gel, then you can say, okay, a high concentration of phosphorylated alpha-synuclein, is that good or bad? It's bad, right? How do you fix that? Lowering the calcium concentration. Okay. Anybody have any questions on this? Does anybody want to move on to the next one? Another good one. Oh, I can say real quick again. Beta actin, right? This is loaded into every sample. This is another control. And it's a control to make sure that you've loaded the same amount of liquid into each well. Okay, so like for instance, if you were to pipette and you pipetted, you know, I don't know, what would it be, like 50 microliters into one well, and then you accidentally did 25 microliters into the next well, right? Then this would have a different, these would show up differently, and then you would know that it was your error and not what's happening on in the experiment. You're like, oh, I messed up pipetting versus, you know, as opposed to this increasing without, you know, getting a false positive, right? If that makes sense. Which is good for your sanity if you're in the lab. Okay, where's this last question? All right, let's try, where are you? The hell? Oh, here it is. Great. Okay, let's do this one. This is another good one. You could take another 3 minutes or so. Anybody got any guesses? Anybody need more time? Got one for C. Give you guys a second more. All right, anybody else? Is there any of these that you'll just eliminate? Someone got A. Someone got D. Someone would eliminate D. Extreme answer. Okay, so with this question, as in with many MCAT questions, do you need to know any of this stuff over here? Is any of this relevant? Absolutely not. Irrelevant. In fact, this drawing is drawing the genome of E. coli, right? And over here, we're talking about plasmids. Okay, so all of this is just stuff to overwhelm you and to freak you out, and we don't need it at all. Okay, this is one of those questions where you just need to know, how a common lab technique works, namely Sanger sequencing and or PCR. Okay, so if I were to tell you that the answer was A, would you guys believe me? Oh, too small. Exactly. Well, the answer is actually A, and because this is a tricky question, and I'm going to go through it. Yeah. So let's read this real quick, right? Table 1 shows blah, 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 linearized plasmids. Okay. So you guys can imagine a linearized plasmid. It's just, a piece of DNA like this. Okay. And so we want to verify that each plasmid contained the selectable marker. Okay. So the selectable marker here is going to be, whatever type of antibiotic resistance is. So there's going to be a sequence for it that we need to identify. And so they want to use Sanger sequencing to verify that the plasmid that they're using in their experiment does in fact have the selectable marker. Okay. And it says that if the thermocycler stopped at exactly 4 cycles, okay, and the reaction started with 1 × 10⁻⁸ grams of linearized plasmid. Okay, so I've got a blank page here. If here's my linearized plasmid, okay, and I'm starting with 1 × 10 to the, what was it, -8? 1 × 10 to the -8 grams of this. Okay. And when I'm drawing my DNA, I always like to draw 5', 3', 3', 5'. Okay, so you guys are all probably familiar with PCR, right? And so the first thing is you would anneal them, right? You have your 5', 3' strand, and your 3', 5' strand. And then what's the next thing you do? You're going to have your primers on them. and they're going to go into the 5 to 3 prime direction. Right? And so after one cycle, you have now two, you have double. Okay, so we went from 1x to 2x, 2 double-stranded segments of DNA, right? And this is times 1 cycle. You guys, this is what you guys all did, right? This is what you're all kind of visualizing. Oops. Yeah. So if that's 1 cycle, then we do another one. You can imagine that we double it. So after 2, we're going to have 4 double-stranded DNAs. After 3, we're going to have 8 double-stranded DNAs. And then after 4, we're going to have 16 double-stranded DNAs. Right? Does this sound right to you? And so if you started off with 1 times 10 to the negative 8 grams of linearized plasmid, right? And that's going to account for both your your double-stranded DNA, and then you end up with 16 double-stranded DNAs. You could maybe conclude that you get 16 times 10 to the negative 8 grams product, right? Is that for people who put C, does that make sense? Is this the same thought process you guys all use to arrive at whatever answer you ended up, whether you accidentally did 1 or 2 or divide by, right? Okay, this would be absolutely right if we were talking about normal PCR. Okay, but we're doing Sanger sequencing. Now, what's the difference between just normal PCR and Sanger sequencing, right? What do we do? What's the critical difference between what happens in Sanger sequencing when you're running a PCR for Sanger sequencing, or just a normal PCR for amplification? Yeah, you're adding the deoxyribose, the dideoxyribose. There's one critical thing here. And that is when you're running Sanger sequencing, you can only use 1 primer. I want that to soak in, right? You can only use 1 primer. Yeah, exactly. So in normal PCR, right, you're just trying to amplify DNA. So you have 2 primers, and these 2 primers are different, so they can go in both directions. Now in Sanger sequencing, maybe I should go back to it. If you were to use 2 primers, then you would resolve both the forward and the reverse direction. So you would actually get it terminating on both things, in both lanes at each base pair resolution. And so you actually wouldn't be able to tell what your DNA sequence is. Okay. And so when we're talking about Sanger sequencing, it's important to know that you're only using 1 primer. So if you were to calculate this again, and maybe I'll cut all this out. How about Sanger sequencing over here? Then you're going to have your 5 prime, 3 prime, 3 prime, 5 prime. And you're only going to have a primer going in 1 direction. Okay. And so after 1 round, you're going to get 1 in the 5 to 3 prime, 2 in the 5 to 3 prime, and then 1 in the 3 to 5 prime. So this is after one round. And so if we were to do this again, right, then you still have this 5 prime to 3 prime. Right, so let's go down here, right? That's still been denatured, right? And then you're going to have the one that was red. I'll just leave it as red. And then you're going to have this one 3 prime to 5 prime. And you're only going to make one single stranded DNA each time. Okay. And so this is after 2. So times 2, you're left with single strand, double strand. And then so after 3, you're only going to add one more times 3, right? You're just going to plus a single stranded DNA. And then times 4, you're going to just only plus a single strand. So at the end, what do you have? You have 5x. I hope this is clear and I can write this out pretty clear. 5x, 5 prime to 3 prime, and 1x, 3 prime to 5 prime, for a total of 6x single-stranded DNA, right? And so remember, 1x double-stranded DNA equals 1 times 10 to the negative 8, right? So divide this by 2 equals 3x double-stranded DNA equals 3.0 times 10 to the negative 8 grams. Sorry for writing over each other.